Hello students, so today we are going to continue to learn the concept molecular basis of inheritance. In today's class we are going to learn about regulation of gene expression, okay. Regulation of gene expression, okay. So this is the concept that we are going to learn today, regulation of gene expression. See students, the genes, they are mainly expressed, ultimately expressed in the form of polypeptides, right. Genes, they are ultimately expressed in the form of polypeptides. So genes, they are ultimately expressed in the form of polypeptides. Genes are ultimately expressed in the form of polypeptides. That means for the genes to be expressed, we have to control them from expressing themselves in the form of polypeptides. So gene regulation can be done at 4 different levels. The first level is transcription of the gene, transcription of the gene, okay. And the second level is processing of the RNA, processing of RNA, okay, processing of RNA. Processing of RNA is nothing but splicing of RNA. Processing of RNA is nothing but the splicing of RNA, okay. And the third level is transport of the RNA into the cytoplasm. See, RNA is formed in the nucleus, okay. RNA is formed in the nucleus. From the nucleus, it has to be transported to the cytoplasm. So, transport of RNA into the cytoplasm from the nucleus is also one more level where gene expression can be regulated. And then the fourth level is translation of RNA. Translation of RNA, that means production of proteins from RNA. These are the different levels at which a gene regulation can be controlled, okay. These are the different levels where the gene expression can be regulated. See students, you know, if you just take prokaryotic organisms, like for example, okay, if we are taking any prokaryotic organisms, okay, if we are going to take any prokaryotic organism, in prokaryotic organism, let us for suppose take the example of a prokaryotic organism, a bacteria which is called as E. coli, okay. E. coli we are taking for example, okay. So this E. coli, Escherichia coli is a bacteria. So generally when when E. coli is growing in the medium called of lactogen, if it is growing in the medium containing lactogen, then from E. coli an enzyme called as beta galactosidase, an enzyme called beta galactosidase is produced. This beta galactosidase catalyzes the conversion of lactose into glucose and galactose. It catalyzes the conversion of lactose into glucose and galactose. Lactose is a disaccharide, students. This lactose is catalyzed to form glucose and galactose in the presence of beta galactosidase, okay. So here we are observing that in the presence of lactogen, the enzyme beta galactosidase is produced by the E. coli bacteria, and then this beta galactosidase, because it is produced in the presence of lactogen, it is converting lactose, okay. It is converting lactose into glucose and galactose, okay. It is converting glucose into it is converting lactose into glucose and galactose, okay. So in this way disaccharide lactose is converted into monosaccharides glucose and galactose as an effect of beta galactosidase. Now if the E. coli is grown in any some other bacteria, in any other medium, if E. coli is grown in any other medium other than lactose, okay, other than lactogen, if any other medium, if it is grown, then what happens? There is no formation of beta galactosidase. Beta galactosidase enzyme is not formed, okay. No beta galactosidase is formed if these organisms were grown in some other medium. So here students, that means it is indicating that beta galactosidase this enzyme is formed only when it is required, right. That means the enzyme which is producing enzyme beta galactosidase is produced only when it is required. That means the gene for beta galactosidase is expressed only when it is required, right. So gene expression is also regulated by this. We understand that gene regulation is also regulated by metabolism, physiological and also environmental factors. Okay, it is affected by metabolism, physiological and also environmental factors. These are the different factors which can affect the regulation, which can affect the gene expression, okay, which can regulate the gene expression. Okay, see students, the next thing is, you know, right from the development of embryo, right from the zygote, we will be observing, right, zygote after fertilization, it will be developing into a complete or new organism with all the body parts, with all the different functions occurring in the body. So see students, when during the development of a zygote till embryo, there are many different genes which are being expressed. So that we will be observing many levels of gene expression occurring right from the development of an embryo till an adult. Okay, during the development of an zygote till the formation of a new baby. Okay, till the formation of a new baby, many different regulations on gene expression will occur. Many different regulations on gene expressions will be occurring. Okay, so in that way gene regulations are found at many different places in our body. According to the different metabolism, physiological and environmental factors, the gene expression is regulated. Okay, so now let us learn exactly how this gene regulation is happening. Okay. You see students, you know, as we have already studied genes, on the genes there are some structural genes. On the DNA, the gene, whatever the fragment of DNA which is undergoing transcription, it will have some structural genes, right. So when we are taking the example of prokaryotes, okay, when we are taking the example of prokaryotes, as we have already studied, in prokaryotes both transcription and translation will occur simultaneously. Both transcription and translation will occur simultaneously. Okay, both transcription and translation will occur simultaneously in prokaryotes. So in case of prokaryotes, at the transcriptional initiation step, initiation step is the main step where the gene regulation can be controlled, gene regulation can be done. Okay, transcription initiation step is the step where regulation of gene expression has to be done. Okay, because after that translation will be definitely occurring simultaneously with the formation of mRNA. So here at the initiation of the transcription only regulation has to occur. Okay, you see students, generally in the prokaryotes, any fragment of the DNA which is undergoing transcription, it will consist of structural genes. Okay, these are those genes which are undergoing transcription. These are those genes which undergo transcription. Okay, it consists of promoter. Promoter is the region to which RNA polymerase will bind. Promoter is the region to which RNA polymerase will be binding. Okay, and there will be some regulator proteins also. There will be some regulator proteins also. Okay, see, these regulator proteins are the ones which are responsible for regulating the expression of structural genes. These regulator proteins, they regulate the expression of structural genes. These regulator proteins, they can be any of these. They can be activators or they can be inhibitors. Okay, that means they can activate the expression of structural genes or they may inhibit the expression of structural genes. Okay, so in that way the regulator proteins are regulating the functions of the structural genes. Okay, regulator proteins are regulating the expression of the structural genes. Okay. And see students, what is the importance of this promoter then? Promoter is the region actually where RNA polymerase is binding, right? Promoter is a region where RNA polymerase is binding and it is going to cause polymerization of the RNA strand. Okay. So now how these regulator proteins are working? We have learned that regulator proteins are activators or inhibitors which activate or inhibit the expression of structural genes, right? So, but how they are working? You know, on the genes there are some regions called as operators. Okay, there are some regions called as operators on the genes. To these operators the regulator proteins will bind. To these operators regulator proteins will bind, and then when the regulator proteins are binding with these operators, it will cause, the effect is gene regulation. The effect is gene regulation, expression of gene regulation. Okay, so in this way the gene regulation is done. So structural genes transcription is done from structural genes. Promoter RNA polymerase will be binding. Regulator proteins will control the functioning of structural genes. They may be activating or they may be inhibiting the functioning of the structural genes. The regulatory proteins, they work by binding with the operators. Regulator proteins, they work by binding with the operators. These operators, they are going to bind to these operators, the regulatory proteins will bind, and when to these operators regulatory proteins are binding, then the effect of the gene expression can be done. Okay, that means it is regulated, or if it is inhibited, or if it is activated according to the type of regulatory protein bound to the operator, gene expression is regulated. Okay, so this is how students gene expression is regulated. You know, we are going to learn about operon concept now. Okay, operon is actually the structural genes plus the promoter and the regulatory and the operator. Okay, structural genes, promoter, and along with the operator the structural genes, structural genes is over, regulatory proteins, regulatory genes. These are all make up an operon. Structural genes, promoter, operator and regulatory genes, these are all going to make up an operon. There are many different operons in the body. Examples: lac operon, tryptophan operon, valine operon. Like that, there are different operons which are known to us. Okay, so let us learn now more about the operon concept. We are going to learn about lac operon, which is seen in the prokaryotes, particularly E. coli. We are going to consider lac operon. Okay, let us learn now about lac operon in detail. Okay students, now we are going to learn about lac operon. Okay, so see students, this lac operon. Lac operon is actually found in case of E. coli. Okay, in E. coli we observe lac operon. E. coli consists of lac operon. So this is a set of genes which is consisting of structural genes, regulatory genes, promoter and an operator. Okay, so it will be appearing like this. So this is how a lac operon will appear, students. So this P is indicating the promoter. for the regulatory gene. I is indicating the regulatory gene. I is indicating the regulatory gene. Okay. So, here I is not a meant inducer. I means inhibitor. Okay. Here the regulatory gene is indicated using I. This means it is an inhibitory gene. Okay. This means that it is an inhibitory gene. From this this means it is an inhibitory gene. From this gene I, repressor is produced. From this gene I, and which is an inhibitory gene, a repressor is produced. Okay. And now you observe the P. P is indicating the promoter to which RNA polymerase will be binding. Okay. O is indicating the operator. Okay. So, here O is indicating operator. Okay. O here is indicating operator. So, to the operator only the repressor will be binding. The repressor produced by the I gene will be binding with the operator only. Now, Z, Y, A, these are all the structural genes. These are all the structural genes which produce different enzymes involved in digestion of lactose. Okay, these are all structural genes which produce different enzymes involved in the digestion of lactose, utilization of lactose. Okay, so Z is a gene which is producing enzyme beta galactosidase. Z is an enzyme producing the gene beta, Z is the gene which is producing the enzyme beta galactosidase. Beta galactosidase is the actual enzyme which is involved in digestion of the lactose into glucose and galactose. Beta galactose is the actual enzyme which is involved in the digestion of lactose into glucose and galactose. Okay, this is beta galactosidase. Then we have another gene Y. Okay, let us write Y here. Y is that structural gene which is coding for the enzyme permease. Okay, permease. Permease is the enzyme which makes the cell membrane permeable to lactose. Okay, it makes the cell membrane permeable to lactose. Okay, and A gene, it is this A gene, it is producing an enzyme transacetylase. Okay, transacetylase. So, Z is producing beta galactosidase, Y is producing permease, A is producing transacetylase. Okay. So, in this way these three genes are produced from the three structural genes are producing these three different types of enzymes. All of them are involved in the digestion of lactose. All of them are involved in digestion of lactose. All of them are important in the digestion of lactose. Okay. So, now we have to see few more concepts related to when is this lactose utilized and all. Okay, now we are familiar with what is a operon consisting of. We have studied that operon consists of a regulatory gene, a promoter or an operator and structural genes. So, regulatory gene is I here, it is going to produce a repressor. Because of this repressor, this is given a name as inhibitory gene because it is producing a repressor which inhibits the functioning of the structural genes. So, it is called as inhibitory gene and it is producing a repressor. P is the promoter to which RNA polymerase will be binding. O is the operator to which regulatory proteins will bind. Z, Y, A is the structural genes. Okay, Z, Y, A are the structural genes which are producing the enzymes important for digestion of lactose. Z is producing beta galactosidase, Y is producing permease, A is producing transacetylase. Okay, in that way these three enzymes are produced by the three different structural genes. Okay, so when these three different enzymes are produced from the structural genes, we have to learn now. Okay. So, now next we are going to see that when, as we have already studied in E. coli, when the E. coli is growing in a lactose containing medium, it is utilizing lactose in growing. When it is not present in the lactose containing medium, it is going to use some other source of obtaining energy. Okay. So, here students, the presence of lactose, switches the structural genes, as on or off. This is the main important thing. Lactose is the one which is switching the structural genes on and off. That means lactose is the one which is responsible for causing the structural genes to be expressed or not to be expressed. Okay. So, how it is responsible for the switching on and off of structural genes? Let us learn now. See students, when lactose is the medium for the organism, okay, when lactose is the medium for organism into which it has been now transferred, in this, when it is in this medium, you know, the effect of the enzymes, permease and other enzyme levels are very low. These levels should be low. Okay, the permease and other enzymes produced from the regulatory genes, they are very low. Under these conditions, when it is growing in the lactose medium and no other source of glucose is available for it, so it is going to take lactose from the medium and try to grow on it. When lactose is taken into the cell, then what happens is, this binds with the repressor. Lactose binds with the repressor and causes the expression of the structural genes Okay, this is the one, this is binding with the repressor and causing expression of the structural genes. Okay, let us pictorially see this now students. This is the importance of lactose medium. Okay, lactose medium enzyme, how is it produced? So, for suppose from this I gene an mRNA for repressor is produced, repressor mRNA, from this a repressor is produced, Okay. So, this repressor, it will always go and bind with the promoter and inhibit the expression of genes. Okay. But now one time what happens, lactose has come into the cell now. Because lactose has come into the cell now, what happens, let us see. Okay. So, now lactose has come into the cell, it is regularly producing mRNA, a repressor is regularly produced, but our lactose, you know, it is acting like acting like an activator for the expression of the genes we have studied, right. So, now the lactose will be coming and binding with the receptor. Generally receptor, it always goes and binds with the operator. Anytime 24/7 repressor is produced, not only when required students, at all the time, every moment in the body of E. coli, repressor is produced. This repressor, when it is produced, it will always go and bind with the operator itself. It will be always going and binding with the operator itself. Okay. But when there is a lactogen, this lactogen is acting like this lactogen is going to act like a substrate. This lactogen is going to go, which is a substrate, lactogen which is a substrate is going and it is going to bind with the repressor. When lactose is binding with the repressor cannot go and bind with the operator. Now, because repressor cannot go and bind with the operator, the RNA polymerase will be passing through the promoter operator and passing through all the structural genes, causing the transcription of all these structural genes, causing the transcription of all those structural genes. So, now when all the structural genes are transcribed, then required mRNA, required proteins, all of them will be forming, and also the enzymes required for the digestion of lactose will be also formed. So, now when the enzymes which are essential for digestion of lactose are formed, continuation of enzyme production will continue until and unless lactose is completely exhausted. Until lactose is present inside the medium, Till then it will be utilizing these enzymes, utilizing the same mechanism and producing more and more these enzymes in this manner. Okay. So here students, we have observed one thing, that binding of repressor to the genes, binding of repressor to the promoter is inhibiting the expression of genes, right. So here there is inhibition of expression of genes. So that is why this lac operon is said to have a negative regulation. Lac operon is said to have a negative regulation. Negative regulation in the sense, lac operon is having a negative regulation in the sense, see, binding of repressor to the gene is inhibiting the expression of genes, right. That because binding of repressor to the gene is inhibiting expression of genes, we are calling it as a negative regulation. Okay. See students, here in this example we have studied that from the regulatory gene repressor is produced, repressor will go and bind with the operator, inhibiting the passage of RNA polymerase onto the structural genes. So no RNA polymerase going on to structural genes, no formation of the enzymes, no formation of enzymes, no lactose is digested, no lactose, no energy. So students, in that way, but the presence of lactose will cause the repressor to be binding with the lactose. Now there is nothing to bind with the operator. Now the RNA polymerase can freely move from the promoter till the last part of the operon. Okay. In that way it is involved in transcribing all the different components of the genes. Okay. In this way RNA polymerase, they will be going ahead into the structural genes, and structural genes will be helpful in translation. Okay. Structural genes will be helpful in transcription of required mRNA and translation of the proteins. Okay. So in this way students, the formation of the different enzymes is occurring only when it is required. When there is no lactose present, then there is no lactose to bind with the repressor as well. So repressor will go freely and bind with the operator, inhibiting all the activities of the structural genes. Okay. So this is how students, the lac operon is working, and in the lac operon there is negative regulation, negative feedback regulation exists in lac operon. Okay. The students, with that we finished to learn completely about the regulation of gene expression. Today we will be learning about few concepts related to human genome project as well. Okay. Human genome project. Okay. See students, the human genome project. So see, you know, everybody is different. Though we are all man, though we are all belonging to same genus, same species, no two people are same, right? And we have already studied that the genetic material is present inside the DNA. DNA is consisting of the genetic material. DNA consists of genetic material. Okay. So it has also been observed that no two individuals are same. No two individuals are exactly same, right? So that is because of differences in their genes. No two individuals are same. 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 Okay. That is because differences in the, because of differences in the genetic material, at least at some points, at least at some points, some points. Okay. At least very small points, there might be differences in the genetic material which is causing the differences between person to person. We are all not same exactly, right? There are differences in their appearance, differences in their behavior, differences in their thinking process. Like that there are many differences between the individuals. So those differences are due to differences in the genetic material. Okay. Those differences are due to differences in the genetic material. So students, based on this concept, a very big project was started in the year 1900s. Okay. In 1900 year, a very big project has been started. Okay. So this is called as Human Genome Project. In short form, it is called as HGP, Human Genome Project. In short form, it is called as HGP. Okay. See students, this Human Genome Project has been started in the year 1990. Okay. So the main intention, the Human Genome Project, what is it trying to do? Let us see. See students, we have already studied that human beings contain nearly 3 into 10 to the power of 9 base pairs, right. It consists of nearly 3 into 10 to the power of 9 base pairs, right. So students, if this is the condition, if 3 into 10 to the power of 9 base pairs are present in the human genome, okay, if each base pair can cost $3 $3, okay. If one pair, for the extraction of one pair, it requires $3. For one pair, it requires $3, okay. Then see, imagine 3 into 10 to the power of 9 base pairs will require 3 into 10 to the power of 9 base pairs will require nearly 9 billion US dollars, okay. It requires nearly 9 billion US dollars, okay. So this is the condition. If that expensive this process is, that much expensive this process is, it is costing nearly 9 billion US dollars, okay. 9 billion US dollars it is costing. And the next feature is, you see, there are 3 into 10 to the power of 9 base pairs, right. So if we are writing them in a book, then it will be like, if there are 1000 pages in a book, if there are 1000 pages in a book, and each page with 10,000 words, each page there will be 1000 letters fitting, okay. Each page there will be 1000 letters fitting. So nearly there are 1000 pages in a book, each page with 1000 words, 1000 letters. Then you know what happens, then it will require nearly 3500 billion books, okay. It requires 3500 million books for providing them the books, okay. It requires many it requires 3500 billion books for recording the genetic material of only 1%, okay. For recording the genetic material of only 1%, this will require nearly 3500 billion such book containing 1000 base pairs each, okay. In that much long our genetic material is. But you see students, for any single person we cannot afford to use 3500 billion books, right. So for that reason there was even involvement of computer technology, which was a budding course at that time, which is called as bioinformatics, okay. Computer technology called as bioinformatics, where we are applying the science of computers to the living organisms. So bioinformatics, from bioinformatics, computer technology's help has been taken, and methods, techniques have been identified to sequence the DNA, okay, put them into proper combination and store the information related to the DNA also. We are having an access, okay, with the help of bioinformatics. With the help of bioinformatics we are having access to computer technology and also the science together, okay. So this is bioinformatics, okay. Now let us see the salient features. Now we are going to learn about salient features. Salient features of Human Genome Project. Salient features of Human Genome Project we are going to learn. See students, the salient features of Human Genome Project is to identify the 20,000 genes to 25,000 genes which are present in the human DNA, okay. 20 to 25,000 genes which are present in the human DNA have to be isolated, okay. And knowing the sequence of nearly 3 into 10 to the power of 9 nearly 3 into 10 to the power of 9 base pairs. Knowing their sequences, what are the sequence of genes that they are having, is also studied under the salient features of Human Genome Project. Okay, and then students, it is helpful in understanding just understanding the it should be able to understand the data analysis, okay, to improve techniques for data analysis, for understanding the data, to improve techniques for data analysis, to improve the techniques for data analysis. Okay, this is another salient feature of HGP. And to store the information, because see, I told you, if we have to store the genetic sequences of nucleotides in 1%, it requires 3500 books with 3000 pages each. 3500 books with 3000 pages each will be required if we are trying to note the sequence of base pairs in a book. Okay, for that reason we need improved technology. So that is, storing the information is also an important concern. Okay, and then students, transfer of this knowledge to other branches, transfer of this knowledge to others. Okay, others, wherever it is required, the knowledge about it has to be transferred. Okay, so transfer of knowledge to the others, these are some of the goals of Human Genome Project. Finally, to address all the social, ethical and cultural issues, social, ethical and cultural issues related to Human Genome Project. So these are all the salient features of Human Genome Project, students. As I have already told you, it has begun in 1900. Human Genome Project has begun in 1900. It ran for up to 13 years. It took 13 years for its completion, and it has got completed in 2003. So Human Genome Project is also called as a mega project. Okay, it is called as a mega project. It is one of the very big very big achievements that human man has got on himself. Okay, that knowing of their own DNA sequences, own gene sequences, own DNA sequences. Okay, so using, students, this Human Genome Project, even a small fragment of DNA, a small piece of DNA can be taken, and the sequence of its nucleotides can be analyzed. Okay, and a complete DNA can be also taken, and its sequence can be analyzed. Okay, so students, analysis of the clear idea of the human genome, understanding the human genome helps in understanding the possible reasons behind the disorders, possible reasons, and treatment and also prophylaxis of many different diseases can be done. Okay, of many different diseases can be done. By using Human Genome Project, by utilizing the DNA of the person, the reasons, treatment and prophylaxis of a disease can be done. Okay, and students, this Human Genome Project was running in coordination with the US Department of Energy, and National Institute of Health, Okay, the US Department of Energy, National Institute of Health, and they were the main leads in this project. And in between, Wellcome Trust was also a very big partner in this project, which was very highly cost effective and which was taking even a very lot of time. Okay, Wellcome Trust was involved. Some other companies like France, Germany, Japan, China, These are the other countries which also participated in Human Genome Project. Wellcome Trust, France, Japan, China and Germany. These are the other countries which are involved in the Human Genome Project. Okay. So, this is all regarding Human Genome Project, students. In Human Genome Project, we have studied the genome of human beings, right. So, the sequence of base pairs in human beings has been studied, but that is not all. Okay. So, the sequence of non-human organisms also, like E. coli, some other bacteria, viruses, plants like rice and Arabidopsis, okay, Drosophila. In that way, many different animals genome was identified using this Human Genome Project. Okay, using this Human Genome Project, the DNA sequences of all the other animals can be also identified, and if there are some of the qualities of the foreign organisms is favorable for us, then it can help us in transforming our own gene expression. Okay, it is helpful to treat many different diseases, cure and prevent many different disorders. Okay, so that is about Human Genome Project. So, students, that is all for today. We have to learn about methods involved in Human Genome Project, of what are its objectives. These are all some concepts related to Human Genome Project are left over. We will continue to learn about Human Genome Project in our next class. Okay, that is all for today, students. We will meet again in the next class. Thank you all for joining the class.